Put that needle down.
All done.
Thank you.
You're welcome. Congratulations.
Thank you very much. Thank you.
Thank you.
Absolutely. All right, I just got my vaccine, and I would recommend it to anyone and everyone. Come with me if you want to live.
This is from Tokyo Metropolitan Government Building Public Relations Section. Tokyo Olympic 2021. It will be February 23, 2021 AD. Since 2020, a mysterious infectious disease with bronchitis and acute pneumonia as the initial symptoms has occurred. It is said that 80,000 corpses have occurred in China, but 1 month later, both corpses and infected people have suddenly disappeared. And then, instead, there is an outbreak in the United States and United Kingdom as well. No matter how much I look for an answer against to this phenomenon, there is no answer from the beginning, nor is there any reason. The only answer I finally arrived at was to make money for vaccination project or Operation Warp Speed. It is reported that highly infectious mutations of the new coronavirus are occurring all over the world, but the number of infected people is decreasing sharply. There is no reason for this phenomenon. However, an abnormal situation, such as a psychic phenomenon and a paranormal phenomenon of synchronicity, are occurring at the same time. Moreover, everything has no meaning. But the problem is that it will continue forever for the future. All that Japan's state of emergency is doing right now is positively shortening the night business hours of izakaya. Izakaya, a Japanese drinking place. Izakaya, Japanese tapas-style restaurant, Japanese bar pub. Japanese bar pub that serves snacks. Rather, the number of Japan peoples going out has increased after the emergency declaration compared to that before. In other words, human thought patterns and behavior patterns are incoherent and uncoordinated. There is no certain regularity. Also, it is unclear what will happen in the future, and there are many uncertainties. More specifically, we don't know what the pathogen is. Also, maybe no one knows what vaccines, such as Pfizer Pharmaceuticals, are. First, the focus will be on whether the thinking and behavior patterns of the government such as China are correct. It is unclear except that the root of infection is human saliva. But now, the new coronavirus SARS-CoV-2 is the pathogen. The mystery is that almost all people infected with this virus are asymptomatic rather and healthy. And the virus is detected in blood samples many years ago. Moreover, a person who has a negative PCR test yesterday may have a positive reaction the next day. There's also the opposite case. How long does it take to get negative once the PCR test shows a positive reaction? No one knows. In other words, at medical institutions, just by adding a respirator, there is no cure or drug, and everything is in a state of luck. As a result, no country does PCR testing for child and worker health checks. However, current science and technology cannot find pathogens by comparing tissue samples of survivors and corpses. Because many patients are hospitalized for pneumonia, the coronavirus may not be the cause, either unreliable or unfounded. In addition, vaccination warp speed operations in U.S. are currently being implemented, mainly in the United States. So, in this class, what exactly is Pfizer's frozen vaccine? Please learn together. Because no one knows what humanity is doing at the global level of the entire this planet right now. I thought now, that is, I needed so important to know it.
Hi, everyone. Thanks very much for having me. I'm delighted to present this webinar and kind of update everybody about some of the formulations that we tried for a self-amplifying RNA vaccine for the prevention of a SARS-CoV-2, or as I call it, SARS-2 infection. So just a general outline of my presentation today. So I'm going to start off with kind of the current challenges in vaccine development and kind of why we chose this platform and what challenges we're trying to overcome. And then give an update on the development and progress of our SARS-CoV-2 self-amplifying RNA vaccine. And finally, talk about some of the future challenges for vaccine manufacturers, specifically in the context of an outbreak or a pandemic. So really the first question we're trying to answer here is, why do we need new types of vaccines? So what's wrong with the platforms that we already have? So really, I sum this up in kind of 6 main categories. So the first is really the lack of efficacious treatment for many devastating infections.
Chemical and
the emergence
biological
of
engineering
multi-drug
from the
resistant
University
bacteria.
of Colorado.
We're always
Boulder.
looking to
She
improve
went
the
on to
safety
do her
profile of licensed
PhD in the
vaccines.
Woodrow Lab.
Obviously, disease outbreaks is quite a hot topic right now. We never know when a new virus will evolve and we'll need to make a vaccine to it. There's also the changing age structure of the population. So we continually have an aging population, so that means vaccines work differently as immune systems change over time. So it's just something that we need to consider for the future. But also, it's always of interest to be able to reduce cost and the time required to produce vaccines. So the two challenges that we're really trying to address with our platform is both the disease outbreaks, so being able to produce a new vaccine very quickly, but also to reduce the cost and time required to
drug
make
delivery,
vaccines.
vaccinology.
So I wanted to give kind of just a general overview of what a nucleic acid vaccine is for those of you that are new to the field. So a nucleic acid vaccine is really just defined as the use of RNA or DNA to express an antigen and then induce an immune response. So I put some just general structures of DNA and RNA on the right there.
In the Michael Smith labs.
The problem with DNA and RNA vaccines is that it requires a delivery vehicle to get the nucleic acid either into the cytoplasm in the case of RNA or the nucleus in the case of DNA, but also to protect it from degradation while it's making that journey.
And she can talk to you about the work they've done on self-amplifying
So there's a number of advantages of nucleic acid vaccines that make them a really cutting edge technology. So on the top, we have kind of this interplay between safety and efficacy. And so it's really been divided into both non-replicating and replicating vectors. So you can see on the right, there's kind of two things labeled there. So replicating vectors as well as live attenuated viruses. And you can see that these are often the most efficacious, but also the least safe vaccine platforms. Whereas on the other end of the spectrum, in the non-replicating vaccines, we have a number of different ones. So non-replicating viral vectors, inactivated viruses, virus-like particles, subunit vaccines, as well as nucleic acid vaccines. And so you can see there for nucleic acid vaccines, they are generally some of the most safe vaccines, but also have kind of more of a challenge when it comes to the efficacy. So the bottom schematic really shows just kind of a general overview of how a nucleic acid vaccine works. So you take genetic material, so a copy of the DNA from the pathogen, and so this is specific to the protein generally on the surface of the virus that you want to induce an immune response to. You put it into an empty plasmid, combine those together using modern cloning techniques, and then from there you can inject the formulated DNA or RNA directly into the muscle. So there's a number of advantages to these. So it's really a speed of development, and I'll show this a little bit later, but the timeline for making a new vaccine is really quite rapid with nucleic acid vaccines. They're also much easier to make than proteins and live attenuated organisms and whole pathogens. It's generally cheaper per dose. So we're generally looking at about $1 per dose when you really start to scale it up into the millions of doses. You can encode any antigen. They're generally safer and less inflammatory. They can be quite thermostable. They enable post-translational modification. So the specific glycosylation patterns that are found in mammalian cells because they're made in human cells, as well as the ability to express membrane-bound proteins. So now I'm going to delve deeper into self-amplifying RNA vaccines. So this is the specific type of RNA that we use. And I think it's really just useful to compare it to both plasmid DNA as well as messenger RNA. So on the left, you'll notice that there's a number of just inherent structural differences between them. So plasmid DNA on the top is a double-stranded circular nucleic acid. For the constructs we use, it's usually around 7,000 base pairs. For the mRNA, it's a single-stranded linear construct, and for our typical construct is around 2,000 nucleotides, whereas the self-amplifying RNA is much larger. So similar in structure, single-stranded and linear to the mRNA, but usually around 9,500 nucleotides. So it's a much, much bigger molecule. However, if we compare it to plasmid DNA, it doesn't require penetration into the nucleus. So it only has to cross one membrane, which makes it a little bit easier to deliver. And there's also no risk of integration into the host genome. Compared to messenger RNA, the self-amplification property, which I'll get into on the next slide, leads to exponentially more copies of the RNA once it's in the cytoplasm, and thus higher protein expression. So it really allows us to minimize the dose that we need for our vaccines. So here's a typical structure of the self-amplifying RNA. So at the top, I have a little more detailed structure of what's actually present. So we have a 5' cap and a poly A tail. These really help protect it from degradation. Also an untranslated region at both the 5' and 3' end. These are integral for the replication of the RNA. We then have these 4 non-structural proteins, and these are what make up the replicase. So this is what gives it its self-amplifying properties. So it's derived from an alpha virus that uses it to replicate the virus's own RNA, and we've kind of just hijacked that system and put it into messenger RNA for our own use. So then downstream of the subgenomic promoter, we have, here I have luciferase, but it's really any gene of interest. So this is where the structural proteins normally would be of the virus, but we've replaced them with either a reporter or a vaccine antigen. So this schematic then shows just the difference between self-amplifying RNA and messenger RNA when it actually gets into the cell. So if you look at the messenger RNA, once one copy is able to cross the membrane, then you have it in the cytoplasm, and that gets translated into a protein. However, when self-amplifying RNA is able to cross that membrane and it gets into the cytoplasm, it's able to activate that replicase. You get many copies of the original RNA and thus much more higher translation of the protein. So the problem here that we've really been working to solve, in addition to kind of optimizing the RNA, is really how do we cross this barrier, get it into the cells, and induce an immune response. So that's more on the formulation side, which is what I'll be focusing on today. So some of our previous work has been in collaboration with Molly Stevens' lab here at Imperial College, where we developed a polymer that we call Pable, which is a bioreducible cationic polymer that we've used for the delivery of self-amplifying RNA. So this is just a schematic to show how it works. So this is the polymer over here. We've made this observation that the higher molar mass Pables that we were able to create in the lab, I worked with a really talented polymer chemist named Frank, who made all of the polymers for us and that we were able to try. So he was able to make them to a higher molecular weight than people had previously. We see that combining them via a titration method or using the PNI instruments, we're able to make really nice uniform particles. And what's really great from this formulation, other than that we see good immunogenicity from it, is that it also enables sterilization. So it fits through a 0.2 micron syringe filter, maintaining its 100% functional RNA through that process, which means that the scale up would be much easier just because you would need to sterilize it before you were able to use it in the clinic. So I just wanted to show a little bit of background data from this. So we've done a number of tests in vivo with this polymer now. And this was just comparing Pable, our polymer, to a commercially available polymer called PEI, which is kind of the gold standard in the field as far as polymers are concerned. So in this study, we were just looking at protein expression using luciferase as a reporter. And on the left, we have it via intramuscular injection, and on the right is via intradermal injection, so into the muscle or into the skin. We compared a number of different groups, so Jet PEI and PEI Max. So these are two versions of PEI. The PEI Max is actually what we use in vitro in the lab for cellular transfections, but it's known to not work as well in vivo. So because of this, we use Jet PEI. And then we used a range of molecular weights of Pable, so everything from 8 to 167 kilodaltons. And you can see here in panel C that we see much higher protein expression from the Pable, although it seems to be kind of this parabolic curve with good expression from the lower molecular weight and higher molecular weight ones, but drops off a little bit in the middle. So we see the same thing for our intradermal injections, although it's really interesting to note that with the Jet PEI, we didn't even see any protein expression with intradermal injection. However, with Pable, we do see really good at protein expression. Again, really good at the 8 and 100 kilodalton levels, but seems to drop off in the middle. And we're still really looking further into what is the mechanism behind this. But because we saw really good protein expression from it, we then moved on to looking at immunogenicity. So one of the models that we have in the lab is as an influenza challenge model. So the antigen that we're using for this is hemagglutinin from influenza, specifically from the Cal09 virus for this study. And again, we compared intramuscular and intradermal injections. So for this, we compared PEI, the 8 kilodalton Pable, and 100 kilodalton Pable. And for these studies, we looked at time points of 3, 6, and 9 weeks. So they received an injection at 0 and 6 weeks, and we gave them doses of 1 and 0.1 micrograms. And what you can see on the left is that the antibody titers that we saw from the 8 kilodalton Pable were higher than what we saw from PEI, which is great. And there's also a dose response. So there's a dose response for both PEI and both of the Pables. Just as you decrease the dose, you see less of an antibody response. For the intradermal injection, what's really interesting is that we actually saw a really poor immune response to Pable intradermally, even though we saw great protein expression. And if you'll remember, we didn't see any protein expression from the PEI in vivo, but we actually do see pretty good immunogenicity from it. So they think this is really just telling us that where you inject it and kind of the drainage to the lymph nodes is an important factor, and we're still kind of optimizing this for Pable. So we kind of looked more into this, and this slide is quite busy, but I'll tell you the main point of it is that we've come to realize that adjuvanting formulations is actually really important. So when we've compared different systemic cytokines, so we've compared Pable, which is the first bar here, and then a number of different LMP formulations at 0.11 and 10 micrograms, we see that Pable is basically completely silent on this front, but the LMPs, especially at the higher doses, are inducing all of these inflammatory cytokine markers. So knowing this, we then start the outbreak of the SARS-CoV-2, or SARS-2 as I've been calling it, started. And so we then really focused our efforts onto making a specific vaccine for the pandemic. So if you're wondering kind of how do you make an RNA vaccine against the SARS-CoV-2, so what we did is basically the coronavirus was sequenced initially in January, and that sequence was made public to everybody when it was published. And so we actually got kind of lucky because the glycoprotein it's very similar to both the original SARS as well as MERS, and so there's a number of conserved regions there. And so because of that, it was pretty easy to work out which glycoprotein was on the surface of this. We were then able to design a pre-fusion stabilized spike protein and encode that in our self-amplifying RNA. We put it into particles, and then we can inject it intramuscularly in mice to test how the immunogenicity goes. So I think what's really interesting to highlight in this is really just the timeline of making the vaccine. So on the 10th of January, the circulating strain was sequenced and was made available to the public. We accessed this on the 20th of January. We then were able to order new DNA oligos, which is how we clone our constructs, on the 22nd of January. Those came in, and we were able to do our gene assembly and coding on the 28th of January. We then made the self-amplifying RNA and were able to transfect it into cells and confirm expression of the spike protein antigen on the 12th of February. So now we're, about a month after the circulating strain was sequenced. We started our immunogenicity studies the next day after confirming that it was sequenced, so on the 13th of February. We started our toxicology study, which is a critical component for starting clinical trials, on the 30th of March. We then were able to submit our paper on the 22nd of April, and then are planning on starting our clinical trials in June. And so, as probably many of you are aware, this is quite actually a rapid timeline for being able to make these vaccines, which I think is one of really the advantages of self-amplifying RNA. So we looked at a number of different formulations in our initial study. So we've actually compared a lot of different things for our SARS-2 vaccine, just because we really wanted to see kind of just what worked best and what we would be able to move into the clinic with. So the formulations that we tried in these studies are Pable, so the polymer that I was just talking about that we've developed in-house. We also looked at adjuvanting it with MPLA. So getting to kind of back to that cytokine secretion and being able to use the carrier as an adjuvant in addition to the RNA. We used electroporated plasmid DNA as a positive control, just because we know this works really well and it's a really consistent across different antigens. And so we use it as a positive control just in all of our experiments when we're making a new type of vaccine. We also used an off-target one, so in this case, rabies, just to make sure we weren't getting any weird signals from the assays or cross-reactivity. But we were also able to directly compare it in our ELISA to recovered COVID-19 patients. So these are samples that we get from the hospital. So we work at the St. Mary's campus at Imperial, and obviously patients are being seen in the hospital. So we were able to get blood from recovered patients and test their antibody and neutralization. So this is an example of some of the antibody data that we got by doing our SARS-2 specific IgG ELISA. So these animals received an injection at 0 and 4 weeks, and we bled them at 2, 4, and 6 weeks. And what you can see is that over time, the antibodies increase, and actually they're pretty similar for as far as total antibody level for Pable, as well as the adjuvanted Pable and electroporated DNA. Obviously, we don't see any background from the rabies control, which is great. But we also see a pretty good response from the patients that we've seen from the hospital, so from the recovered patients. But what's really interesting is that the vaccine actually seems to be, in mice at least, seems to be working then better than a natural infection, which was really encouraging for us. So the second aspect to this is not just the antibody titer, but how functional are those antibodies. So we were able to develop pretty rapidly what's called a neutralization assay. So we made a pseudotyped SARS-2 virus, and we're able to look at just how well the antibodies prevent the virus from entering cells, or I guess the pseudotype virus in this case, and then again compare it to the recovered COVID-19 patients. So on the y-axis here, we have the IC50 of neutralization. And then you can see for each group, so we did this for the 4 and 6 week time points, so just as the antibodies were starting to come up. Again, we don't see any cross-reactivity from the rabies control, so we can be really confident in this data. Again, the Pable and the adjuvanted Pable worked quite well, but very similarly, as did the electroporated DNA, which is great. And again, with the vaccine, we're seeing even slightly higher neutralization for those antibodies than we did for the recovered patients. So this was all really encouraging. Another thing we wanted to look at specifically for this indication is the Th1, Th2 skewed response. So if you're in the field, there's been a lot of talk of this antibody-dependent enhancement, which usually happens historically with SARS and MERS in a Th2 vaccination. Or I guess when a vaccination skews it towards a Th2 response. So this is something we wanted to check. So we did the ELISAs for the IgG1 and IgG2A to test this, which is presented here on the left. So again, it's just the amount of specific IgG in the sera. So you can see on the right, when we compare the skewing, so if you do the ratio of IgG2A to IgG1, this is the Th1 to Th2 skewing. So if it was right at 1, it would mean there was no skewing. If it's above 1, that means it's Th1 skewed, and if it's below 1, it means it's Th2 skewed. So what's actually really great and also pretty expected for RNA vaccines is that we are seeing a Th1 skewing here. That ratio is about 10 to 1, which is great for these data. So probably the next question that people always have is when will the vaccine be ready, and just a little insight into the clinical trials. So we are now pretty much done with all of our preclinical animal studies. So our toxicology study is finished. We are just kind of in the final stages of analyzing that histology data and then being able to submit all of our paperwork to the regulators, which is really exciting. This is our current plan for our clinical trials. So we're intending to start off with a dose escalation starting in June. That will be 40 people. We'll then move into a safety and immunogenicity study of 100 people, and then really into our bigger trials. So the efficacy studies are planned for September to January of 2021 at this point, which is set to include 5,000 people. And then pending how all of that goes, finally the implementation for 20 million people. So this is quite a rapid timescale, which also really brings into question of how quickly can you scale up your vaccine, and is it actually possible to make 40 million doses to treat 20 million people. So in the meantime, we've been continuing to hone our formulations. As I said, we've tried a number of different things. And what's really exciting actually is that we have started some work with PNI, so using some of their lipid nanoparticle formulations. So this wasn't a SARS-2 study, but rather an influenza challenge study. And we did this just because it's a really robust model, and you can look at a number of different factors of the immunogenicity. So on the left here, I just wanted to present some kind of preliminary data. So on the left here, we have the hemagglutinin, so the flu-specific IgG as tested by ELISA. These animals got injections at 0 and 4 weeks, and then antibody ELISAs at 4 and 6 weeks. And we compared it to our positive control. In this case was Pable, because we know pretty much how it works now. And then 2 lipid nanoparticle formulations from PNI. So what was really exciting about this is you'll notice that for most of the groups, there's 5 points. But for this LMP1 at the 6 weeks, there's only 3 points. And that's because for a normal ELISA, we do a 1 in 100,000 dilution. And that usually pretty much captures all of the antibodies that we're going to be able to detect in the linear range. But 2 of them were out of range above the limit of quantification for this assay. So we need to go back and repeat them. So that was exciting because that's actually never happened to us before, but it's obviously a good sign that we need to dilute it even further to detect it. And then on the right is another aspect of that challenge study. So when we challenge the mice with flu, one of the main markers here is their loss in body weight. So this is kind of the way that you can compare formulations in addition to the ELISAs and see how well they're working. So as you can see here, the naive mice lose a significant amount of body weight and have to be cold, usually between days 4 and 6, which was also true for the study. So it's a good control to show that the challenge was working. You can see here with the Pable, where we're seeing pretty good antibody levels, we still see some weight loss, about 10% at the peak of it. But with both of the PNI formulations, we didn't see any weight loss with the mice, which is, pretty much what we were expecting giving these ELISA results. So seeing really high antibodies pretty much means that they should be completely protected. So we're really excited about these formulations and moving forward. I kind of wanted to finish up this presentation with just thinking about how do we make vaccines and how can we do this in a better way. So something that I think is really interesting is that if you think about most of the vaccines that we get now, which are generally protein and viral vaccines, like I talked about in the beginning, this is a picture of making the flu vaccine in 1957, and this is also a picture of making the flu vaccine in 2020. And you'll notice that we still do it in eggs. It's not a very high throughput method, and that really limits the number of batches of flu vaccines that we can make per year. And so it's a very archaic technology, and I think just could really be improved upon. But in addition to that, I mean, other vaccines are made in kind of these huge bioreactors, right? So when you're trying to scale up doses for millions of people, you're really just limited by cells growing in a container and having access to these huge containers to grow this in, and it's really limited by time. So what I think is really beautiful to demonstrate about specifically self-amplifying RNA vaccines is that for the doses that we're going into human clinical trials in, we can make 100 milliliters, 1 million doses. So this really reduces the footprint that you need to be able to make these, right? So we don't need a whole manufacturing facility. With the appropriate amount of money, I could make 1 million doses in the lab because we can make 100 mil batch. And so what I think is really useful is actually these PNI systems. So we just recently got our new Ignite, but it makes it so that you could start to think about locally producing these vaccines, which I think will be more and more important as, different countries and places all over the world think about making vaccines for pandemics, but also just kind of, different indications in general. And so, I kind of wanted to leave off with what I think will really be the future of manufacturing of vaccines. And so for now, as far as our clinical trials are going, we are getting, there's these, 4 different places where we're getting main components of it from. And this is actually kind of rapidly changing even as we scale up. But at the minute, we get the RNA made in California. We then are getting lipids from Vancouver. Those get combined in Austria in a GMP formulation unit there. They actually then get shipped to Italy to be put into vials and then sent to the UK for the clinical trials. So this involves a lot of coordination and, is really difficult actually to ship this all over the world when things need to be a certain temperature and it can only last so long. And it's quite an ordeal. So I think really what we're going to move forward towards is really kind of these local manufacturing centers. So just because you can do it on such a smaller scale and make lots of vaccines, as long as you can do it to GMP, you'd be able to do it locally and produce vaccines for people in certain countries. So I think instead of having these major manufacturing centers, we'll start to see more and more smaller ones where you can do the technology kind of all in one place, make the RNA, formulate it, and then put it in vials. So this is kind of more of what I was envisioning, although you'll notice that I got tired of copy and pasting this somewhere around the Middle East. And so there's much fewer in China and Australia than probably would need to be. So in conclusion, as far as the immunogenicity go, we saw that our self-amplifying RNA vaccine induces high antibody titers, higher antibody titers and neutralization than a natural infection from recovered COVID-19 patients. We're starting to look at some next generation formulations. So this includes the PNI LNPs, which we've seen to have enhanced immunogenicity of the self-amplifying RNA even compared to Pable. For our clinical trials, we're set to begin our phase 1 in June 2020, so coming up in a few weeks, which is really exciting. And I think as far as the future challenges, we're going to start to see more of this modular manufacturing of RNA that will really enable a global response to future outbreaks, so we won't be waiting on kind of major pharmaceutical companies, et cetera, to be able to do that. So with that, I'd like to acknowledge all of the people that were involved in this work. So at Imperial College, so specifically Robin and Molly, Paul and Frank, who worked on this work, as well as the rest of the team. It's been a very collaborative, exciting effort. And so, yeah, it's been a really, really cool time to be working on this. As well as our collaborators at the Karolinska Institute, as well as in the NHS Imperial College Trust.
All right, so I think with that,
for the clinical trial. So this involves a lot of coordination and, you know, is really difficult actually to ship this all over the world when things need to be a certain temperature and it can only last so long, and it's quite an ordeal. So I think really what we're going to move forward towards is really kind of these local manufacturing centers. So just because you can do it on such a smaller scale and make lots of vaccines, as long as you can do it to GMP, you'd be able to do it locally and produce vaccines for people in certain countries. So I think instead of having these major manufacturing centers, we'll start to see more and more smaller ones where you can do the technology kind of all in one place, make the RNA, formulate it, and then put it in vials. So this is kind of more of what I was envisioning, although you'll notice that I got tired of copy and pasting this somewhere around the Middle East. And so there's much fewer in China and Australia than probably would need to be. So in conclusion, as far as the immunogenicity go, we saw that our self-amplifying RNA vaccine induces high antibody titers, or I guess higher antibody titers and neutralization than a natural infection from recovered COVID-19 patients. We're starting to look at some next generation formulations. So this includes the PNI LNPs, which we've seen to have enhanced immunogenicity of the self-amplifying RNA even compared to Pable. For our clinical trials, we're set to begin our phase 1 in June 2020, so coming up in a few weeks, which is really exciting. And I think as far as the future challenges, we're going to start to see more of this modular manufacturing of RNA that will really enable a global response to future outbreaks, so we won't be waiting on kind of major pharmaceutical companies, et cetera, to be able to do that. So with that, I'd like to acknowledge all of the people that were involved in this work. So at Imperial College, so specifically Robin and Molly, Paul and Frank, who worked on this work, as well as the rest of the team. It's been a very collaborative, exciting effort. And so, yeah, it's been a really, really cool time to be working on this. As well as our collaborators at the Karolinska Institute, as well as in the NHS Imperial College Trust.
我们今天要介绍的一个主题就是 DNA 的剪刀手 CRISPR-Cas。那什么是 CRISPR-Cas？Hello,
my name is Ana de Molins, and I'm going to highlight some aspects of my review work named Perspectives of Nanocarrier Drug Delivery Systems to Overcome Cancer Drug Resistance in the Clinics. So, as you know, cancer is one of the leading causes of death in the world. Specifically, in 2018, in Europe, there were nearly 2 million deaths caused by cancer and 4.2 million new cases diagnosticated. Even though there has been a progress achieved in the development of more effective therapies against cancer, recurrence of tumor growth and metastatic spread in the long term is a common event due to the increased resistance. Therefore, a closer understanding of the mechanisms involved in drug resistance is needed. Cancer cells are masters in finding a way to resist the treatment. The different mechanisms are shown in this figure. First, and the most studied, is elevated drug flux, where due to the expression of the ABC transporters, which are ATP binding cassette proteins, cancer cells push the substrate out of the cell and decrease the intracellular drug concentration, acquiring multi-drug resistance. In B, we see unknown mechanisms of cells that consist of simultaneous metabolic reactions to finally expel the harmful molecules out of the cell via the ABC transporters mentioned in A. Also, it is possible that some of drugs are unable to interact with their molecular target, even if it's a surface target or an intracellular target, because it has been altered by mutations or modifications. In D, it is known that repair pathways known as DNA damage response are overexpressed of cancer cells. So, when a drug affects the DNA of cancer cells, producing a DNA damage, they can repair it due to the expression of these genes. Linked to this In E, it has been described that cancer cells have changes in apoptosis-related proteins to avoid the programmed death. All the mechanisms mentioned before come from the epigenetic cancer adaptation. These mutations lead to genomic instability and generate a great level of genetic heterogeneity within the tumor. Therefore, a tumor contains various types of cells and extracellular matrix known as tumor microenvironment, as we can see in the figure. It is composed of differentiated cancer cells, cancer stem cells, normal stromal cells, fibroblasts, mesenchymal cells, and tumor-infiltrated immune cells. It also includes several soluble factors, such as cytokines and growth factors. All of these components within cancer cells orchestrate a complex dynamic network with a common objective: survival and spread. Patients with recurrent resistant cancer show higher number of cancer stem cells within the tumor. These cells share several properties of stem cells like cell renewal, tumor initiation capacity, and long-term repopulation potential. Its importance relies in the inner capacity of resistance to drugs and toxins through the expression of different mechanisms that I mentioned before. They also can survive in non-attachment conditions and can initiate a tumor growth in vivo, leading to an increased capacity to migrate, intravasate, and generate metastases. It is important to target them as well as all the components of the tumor microenvironment to be able to overcome cancer recurrence. Nanotechnology-based drug delivery systems can incorporate anti-cancer drugs that have poor solubility, low bioavailability, and inadequate toxic profile. Among them, nanoparticles have the ability to transform conventional medicine by reducing the adverse effects and providing a controlled release of therapeutic agents. There are different requirements in nanoparticles to be used as delivery systems that include size, biocompatibility, and surface chemistry. Explain in more detail in my review. Ideally, the synthesized nanoparticles must remain stable in the bloodstream until they reach the cancer cells within the tumor microenvironment. The nanoparticles can be made from a variety of materials. Today, they are classified into 5 categories shown in this table. There are lipid-based nanocarriers, polymer-based nanocarriers, drug conjugates, inorganic nanoparticles, and viral nanoparticles. They have different advantages and drawbacks depending on their application, so a proper study is needed to select one for the desired application or purpose. The first nanoformulated drug approved by the FDA was Doxil to treat Kaposi sarcoma in patients with human immunodeficiency virus in 1995. It is composed of doxorubicin encapsulated in liposomes, and it showed a reduced cardiotoxicity compared with a conventional drug. Nowadays, it is widely used to treat the original indication and other types of cancers. Furthermore, another interesting clinically approved nanomedicine is Abraxane, a formulation of paclitaxel complexed with albuminum-bound nanoparticles that was approved by the FDA in 2005 to treat metastatic breast cancer. It shows a substantial reduction of toxicity because paclitaxel is high hydrophobic and has to be diluted in an oil that has systemic and neurological toxicity. So the increased tolerance of Abraxane, which does not use this oil known as cremophore, implies better safety profile and allows administration of higher doses of paclitaxel. Regarding the combination therapy, Vyxeos was the first dual drug liposome approved by the FDA in 2017. It is a liposomal nanoparticle for the treatment of acute myeloid leukemia that incorporates 2 drugs, cytarabine and daunorubicin, in an optimized ratio. The major advantage is the possibility to target 2 different disease pathways and reduce the side effects. Nanoparticles are in constant development, and in Table 3, you can see some of the clinical trials that are taking place at the moment with different types of nanoparticles. The majority of nanoparticles in tables 2 and 3 are passively accumulated at the tumor site. Passive targeting is a non-specific accumulation of the nanoparticles in the cancer tissue. It is possible in solid tumors where there is an enhanced permeability and retention effect. This effect is due to the specific pathophysiological characteristics of the tumor vessels that are generated by angiogenesis to provide nutrients to the malignant cells. The abnormally wide fenestrations in these blood vessels due to the production of vascular permeability factors and the lack of lymphatic drainage facilitate extravasation of the nanoparticles. One of the main challenges is to engineer a targeted drug delivery system capable of specifically binding cancer cells and avoid the non-cancerous ones. The principal factors that control this targeting are the surface functionalization on the nanoparticles, their physicochemical properties, and the specificity of the target moiety, and the pathophysiological characteristics of the tumor microenvironment. Different clinical trials with targeted nanoparticles are taking place at the moment, shown in this table. Even though promising preclinical data regarding the use of targeted nanoparticles has been achieved, the clinical outcomes are still modest, and there are several reasons for the limited clinical translation of these cancer nanomedicines. One of them, for example, are the engineering limitations, because many methods of functionalization are published, but most of them lack reproducibility. Also, it is a very complicated process with multi-steps that compromises the final production yields. Moreover, even all this exhaustive process, obtaining uniform size is not yet a reality. Due to the process variation, there is a lack of scalability that prevents large-scale manufacturing. Further complication is presented by the biological conditions of the tumor. Even though nanoparticles extravasate into the tumor vicinity, targeted nanoparticles commonly bind cells with high affinity in the outer layer of the tumor microenvironment, known as the binding site barrier, which prevents the deeper penetration of the nanoparticles. Also, there is an overestimated EPR effect and the existence of elevated tumor interstitial fluid pressure that reduces the delivery of the tumors. There is a wide variety of nanomaterials available, and their physiochemical properties play an important role in the activation of an immune response. Moreover, the introduction of a targeting ligand changes the properties of the nanoparticle and often makes them even more difficult to pass unnoticed. The screening for immunotoxicities are insufficient to address the broad spectrum of biomarkers that indicate nanoparticles immunotoxicity. In addition, there is an absence of consensus on well-characterized reference materials. Therefore, preclinical studies often depend on nanomedicines with known clinical immunotoxicities. The use of immunosuppressed animals makes it even more difficult to determine immunotoxicities related to nanoparticles. And similarly, in clinical studies, patients are pre-medicated with immunosuppressors to prevent adverse reactions. And finally, there are financial challenges that hamper the development of new nanoformulations, because it is not easy to demonstrate improved efficacy and safety compared to other validated and marketed products for the same indication. Indeed, the majority of approved nanodrugs are based on currently approved drugs, which faces a reduced financial risk. But in more complex nanodrugs that contain new chemical entities, there are economic considerations involved. Also, the design of a specific ligand and the conjugation technique makes the whole process more expensive with a difficult scale-up. Apart from all the high costs involved, regulatory requirements also make market entry difficult. The EMA and the FDA start their discussion on the classification of nanomaterials and how to regulate them to ensure the proper efficacy and safety of these materials. All the mentioned drawbacks translate into poor clinical outcomes, particularly of targeted therapies. Therefore, there is a clear need to focus on ways to enable rapid and more efficient clinical translation. Hydrogels, for example, are formed through the cross-linking of hydrophilic polymer change, and their water-rich nature makes them widely applicable, including for drug delivery purposes. The advantage against active and passive nanomedicines is that they can provide localized and targeted therapy regardless of the bloodstream and the microvasculature morphology of the tumor. Another interesting alternative is to learn more about nature's own delivery systems. Exosomes are a class of extracellular vesicles that contain proteins, nucleic acids, and lipids. They are the key players of numerous biological processes, both pathologic and non-pathologic. In cancer, specifically, exosomes are mediators of tumor-stroma interaction. This crosstalk is known to be involved in migration, treatment resistance, and metastasis. This opens a window for target metastasis, which has not been achieved with the conventional nanosystems. Also, these natural drug delivery systems can pass unnoticed by the immune system and can transport classical chemotherapeutics and nucleic acids to treat cancer.